Okay, so last time we spoke about gastric emptying, and basically what I mentioned was there's this kind of complicated dance between the stomach and the intestines where the duodenum really tells the stomach what it can handle as far as the amount of chyme it can receive and how often. So we've all experienced this, whether or not we knew it. I'm sure we've all had times in our lives when we've eaten a meal and felt like we needed to eat again really soon after. So that's an example of rapid or fast stomach emptying versus a time when you ate something and you just felt full for hours and hours and hours. And you're like, man, is this meal ever going to end? So that perception of fullness and the length of fullness is related to the hormones that you see before you and how they're released basically in pulses. So I'll show you the feedback loop in a moment here. So let's start with gastrin. It's secreted by G cells when they are exposed to indigested proteins. So basically this is a trigger. The indigested proteins are a trigger to say, hey, we have a ways left to go as far as getting amino acids out of our food. So we need to stimulate the stomach to do more of that work before we receive the chyme. So that's really the goal there. And if you remember the hormones and also the enzymes of the stomach, that should make sense too, because, I'll grab my little laser pointer here. Remember, the stomach produces pepsinogen, which is converted to pepsin by hydrochloric acid. So if gastrin is released, it's going to increase the rate of release of the two things that really help proteins get broken down. Secretin is the next one. So this is when any chyme arrives in the duodenum. And this primary effect is to trigger buffer and bile release. So your stomach acid varies in its intensity, depending on what meal you ate, depending on how recently you ate or didn't eat. But it's always going to be more acidic than the duodenum. And so any amount of chyme entering the duodenum is going to trigger the release of secretin because you need to buffer that acidic chyme. The cells of the duodenum are not designed to withstand that low pH. So the tubular cells of the pancreas, remember it's the ductal cells that release that bicarbonate, and bile by the liver are going to be released. And the secretin also is going to reduce gastric motility and secretory rates. Gastric inhibitory peptide does exactly what it sounds like. So basically this hormone is going to press pause on stomach emptying while the duodenum and the pancreas have time to deal with the chyme that they've received, especially if that is really rich in fats and carbs, which take a little bit longer to digest. Cholecystokinin is secreted when chyme arrives as well, but particularly when the chyme is dense with lipids and proteins. So a really good example of this would be if you go out to a fancy steak dinner, you would expect to see a spike in cholecystokinin compared with, say, only eating a salad, right? So cholecystokinin has a positive feedback effect on the production and secretion of a bunch of other things, and we talked about that as well. So its effect on things like chymotrypsin, trypsinogen, etc. But sort of the broad and overall effects is to really push for enzyme secretion. So there's this big push to increase the rate of enzymatic hydrolysis of whatever is in the duodenum. It also inhibits gastric activity. And this last sentence appears to have, is your book's nice way of saying, there is mixed evidence of this, so we're not going to take a hard stance on it. We're going to say maybe it does this. But there is some research to suggest that if you, say, administer cholecystokinin experimentally, impulses to eat diminish. Now, exactly where in the CNS that works specifically is unclear. Probably in areas involved in appetitive eating behavior, but nobody knows for sure. So just know that whenever your book says appears to or may, it means that no one knows for 100% sure, but there is some research that says yeah. Vasoactive intestinal peptide we didn't really talk about, but if you remember that all of the juice that enters your GI tract is watery and therefore requires blood filtrates, that's what this does. And it also helps with absorption. And then enterocrinin is basically all about mucus. So you want to make sure that there's adequate mucus protection of the duodenum from the nasty acidic stuff coming in from the stomach. So here's a summary of those relationships. And this would be a good figure to have on hand when you're studying and when you're test taking. Because let's be perfectly honest, we have limited time left and you have limited more information that you can conceivably cram into your overworked heads. I totally understand that. I want you to come away from the class with a healthy understanding of the regulation of stomach emptying, but I don't expect you to have 100% mastered all of this by the time you leave. I don't think that's a reasonable expectation. I would love it if it were true, but you got a lot of stuff to know, and I get that. So what I would recommend is taking this slide and its figure and just having it on hand and handy. Because here's the thing, with timed tests, if I ask a multiple choice question about this, and I'm very likely to, if you've done enough studying to be passingly familiar with these things, what that means is even if you can't answer the question by yourself right away, you should be able to use this chart to answer it pretty quickly without having to do much searching. And that's about as much mastery as I need for you to show me. Does that make sense to everybody? We're kind of splitting the difference here between being like a gastroenteral expert and having enough knowledge to understand why your stomach empties at the rate that it does. Okay. So I was sitting eating dinner with my partner last night, and he was like, "What are you teaching tomorrow?" And I was like, "Well, I am teaching the end of the gastrointestinal system and the beginning of urinary system." He was like, "Oh, so poop and pee." And I was like, "Yep, poop and pee, indeed." So that's really what we're doing today. So the large intestine begins with the cecum, which is this pouch-like appendage at the bottom here. And here you can see the ileocecal valve that regulates the passage of feces into the cecum. The appendix, if you have one, hangs off of the cecum. I don't have mine. Mine got really angry when I was about 16 and had to come out. Some people never have that problem. It depends. The ascending colon refers to the area of the colon where feces is ascending. So a lot of these are just named for the direction in which the feces is traveling. Transverse is transverse. Descending is descending. And then the last little bit, the sigmoid, is not named for direction, but rather its shape, so S-shaped. And then after this flexure here, so this little area where I'm hovering my laser is the area where the colon becomes the rectum, and then down here is the anal canal. And the anus is much shorter in length than most people believe. It's only a couple centimeters. So the large intestine, which is also called the colon, and that's an important note because sometimes students get the two confused, or they'll think that the colon means the large intestine that I just showed you, and then the large intestine means small intestine. It's one of the drawbacks of having there be so many terms for a lot of these things. They have a lot of stuff in anatomy, has a lot of names. But large intestine is the thing I just showed you. It is synonymous with the colon. So with regard to the physiology, which is to say, why is it there? Especially why is it there if the majority of nutrient absorption is performed by the small intestine? Well, a couple of reasons. One is your small intestine, and I'll show you a figure that shows this a little bit later. Your small intestine contributes a tremendous amount of water in the form of its secretions. That water comes from your blood. You want to get that water back. So large intestinal function is really concerned with absorption of water and also whatever ions are suspended in that water back into your blood. Vitamins as well, and bile salts. So you can recycle the bile salts. So if you think back to the fat emulsification slide that I showed you, those bile salts, which are made out of cholesterol and which perform important emulsification functions, you can actually reuse those and your liver will recycle them. So you take those back as well. So let's talk about coliform bacteria. In in-person classes, I typically use this slide as a jumping off point to show a video, which I'll explain in a minute, but I don't think I can in this particular context because it's a video that I didn't make and it's on Vimeo and I don't know if I have permission to reproduce it in a recording. So instead, I will tell you where to find it if you want to watch it, and I'll let you decide if you want to. So, to frame my argument for watching this video, interestingly, you have a lot of bacteria in your colon, they're called coliforms, that are either harmless and you never notice them, or that basically pay rent. So you have tons of bacteria in your colon, basically getting free housing and food. So they get food in the form of the food you eat, and they get free housing in the form of they get to live and reproduce in your colon. So you have all these little vagrants living in your colon. Some of them are freeloaders and don't do anything for you and just hang out in there. Others of them actually pay you rent by making things for you, which is pretty cool. So one of the biggest deals with the ones that pay rent is the bacteria that make vitamin K for us. About 50% of the vitamin K that you have circulating actually comes from bacteria making it for you, not from diet. And if you think back to the blood chapter, at the end of the blood chapter, we discussed clotting. And the clotting factors, of which there are tons, remember that big chart? There's a table in your book that shows all of the clotting factors. So, factors 2 through 12, including factor X, which starts off the common pathway, etc. Most of those were synthesized in the liver and required vitamin K for their synthesis. So in a very real sense, some of your little intestinal tenants are the very creatures that allow you to clot your blood because they make an important vitamin for you. Also biotin. Biotin is a vitamin that helps with epithelial growth. So if, for example, if you go to like a GNC or a Walgreens or some drugstore, you can purchase bottles of biotin as a supplement, and it'll say something on the bottle like hair, skin, and nails. Biotin contains building blocks for healthy keratin and promotes mitosis at the bed of nails and the root of hair. So it's basically all about having a healthy epidermis and epidermal derivatives. And then vitamin B5 is just one of the many B vitamins that exist. So although the idea of having bacteria live in the colon disgusts many, you actually need it to be healthy. So the video I mentioned. In the last 2 decades or so, fecal transplants have gained popularity both medically and in the news. And if you're wondering if I really mean transplant of poop from one person to another, the answer is yes. So the video I want you to view, if you've got the time, and it's not gross, by the way, it's a short documentary, it's on Vimeo, and you can find it really easily if you just type into Google, why would anyone get a fecal transplant? So the video concerns a young woman who suffered a very bad staph infection, and in the course of treating the staph infection had to take really heavy-duty antibiotics because she had MRSA, methicillin-resistant Staph aureus. So one of the consequences of her staph treatment was that it killed almost all of her intestinal bacteria. Inside all of us, we have a bacterium called C. difficile, or C. diff, that would very much like to take over our colon, but it's suppressed by the rest of our healthy gut flora. So it's basically waiting in the wings to cause harm if the opportunity arises. So in this poor young woman, the opportunity did arise. So vancomycin and other heavy-duty antibiotics wipe out the rest of her gut bacteria, but her C. difficile keeps popping back up. And if you work in a clinical setting and you've ever smelled the smell of C. diff, it's not one that you forget. It's pretty gnarly. So the patient in question in this video, she and her parents describe the impetus for battling with C. diff and getting the fecal transplant. And then doctors discuss it, as well as the challenges for getting it fully FDA approved, because there's questions around regulating fecal transplant. Like, do you treat it like an organ? Do you treat it like blood donation? Like, how do you classify it? How do you make laws about it? So it's really interesting. And basically the idea there is to take some feces from a healthy person that ostensibly has healthy bacteria and use it to seed a new community of healthy bacteria in the recipient. So go watch it if you're interested. It's pretty fascinating. All right, so back to details about the large intestine. Excretion of organic waste is one. So urobilin and stercobilin are bile breakdown products, and these also happen to give feces its color. Indole, scatol, ammonium ions, and hydrogen sulfide, these are all consequences of amino acid and peptide breakdown. These are what account for the odor of feces, specifically indole and scatol. Hydrogen sulfide has a more sulfury smell, so it's more the smell of flatus or farts, but the actual smell of feces is more about these two volatile hydrocarbons. Mass movement, which is just the mass movement of feces within the colon towards the rectum and anus, is stimulated by the distension of the stomach and the duodenum. Okay, defecation reflex. So the defecation reflex contains mostly involuntary components. So as we all know, there's a portion of defecation that we all have control over so that we can choose an appropriate time and context in which to do this. So hopefully the bathroom and in private. But there's also a bunch of stuff that goes on in the background that is not voluntary and is basically autonomic. So just to give you a sort of overview here, the purplish arrows are the long reflex and the bluish arrows are the short. So like many things in the GI tract, there is an interlocking sequence of short and long reflexes that regulate all of this. So here's the flexure after the sigmoid colon, which is somewhere over here. Basically, when you get the arrival of feces in the rectum, it pushes on the walls of the rectum. And that stimulates stretch receptors. So that's our start point. That's the start of all of this stuff. So in the short feedback loop, we've got stretch receptors, which also innervate the myenteric plexus, which remember is that independent network of nerves in the muscularis externa. So they experience an increase in tone when they are stretched, and that causes increased peristalsis just in this region, so pushing feces towards the anal canal. Stretch receptors also kick off the long reflex, but this is mediated via parasympathetic nerves in the sacral spine. And remember, we have parasympathetic innervation of smooth muscle. This fits the bill. These, in turn, help to stimulate somatic motor neurons. So if you pay close attention to this leg of the long reflex, this is when you start to have the option of voluntary relaxation of the external anal sphincter, which is part of the pelvic floor. So this loop of the arc is the part that you have control over. The rest of this, nope. So the other outcome is increased peristalsis throughout the entire large intestine. And both of these, so local and broad peristalsis, are going to continue to move feces into the rectum and basically amplify these two loops in a short bout of positive feedback until you voluntarily relax the anal sphincter and defecation is complete. So, time frame for digestion. Oh, one thing I wanted to say before we move on, because this is something that might come into relevance for some of you during your careers. In patients who have experienced spinal injuries and lost control of parts of this feedback loop, so for example, home care nurses and people engaged in certain fields of occupational therapy, especially if you work with paraplegics. In paraplegics, when parts of this loop are broken, there's interruptions in these feedback loops, and those people require assistance with defecation because their reflex arcs are broken. So typically that requires stimulation of this area, either manually or with drugs, to cause defecation to be able to be complete. And that's really important for the health of and well-being of those individuals, because bowel impaction is a very real risk there. All right, time frame for digestion. Swallowing food, 4 to 8 seconds if you're swallowing a bolus, 1 to 2 seconds for liquid. About 4 hours, I should say hours, not horse, for an average meal. But again, high-fat meals take longer because that means that the duodenum is going to release hormones that inhibit digestion at a higher frequency. The transit time in the small intestine should be about 2 hours. And this is important because this is what gives adequate time for absorption. And the large intestine is longer, so 12 to 24 hours. So this is the time frame on average that is going to be healthy and prevent diarrhea or constipation. Okay. So now we're getting into some of the slides that ended up being hidden. So if you don't see these, if you're following along, don't panic. You just have to go unhide them. So this is, the next few slides are just basically going to focus on the difference in breakdown and absorption between lipids and the other macromolecules of nutrients. So remember, lipids need to be emulsified as well as undergo enzymatic hydrolysis. So both of those are important. So lingual lipase is where that starts. So remember, lingual lipase comes from the saliva, and it is an ACE, meaning an enzyme, so that's going to break triglycerides up into monoglycerides and fatty acids. Monoglycerides and fatty acids need to be emulsified, and they get organized into micelles in the intestine. So a micelle is just a tiny, tiny little ball of lipids broken up with bile salts. And then I know that this box is blocking this, but I'll tell you what it says. So micelles are absorbed into the intestinal epithelium. That's not very difficult because, of course, they're lipid-based, and so are the phospholipids that make up the bilayer here. So with regard to absorption, it's pretty easy to absorb fats, and they get broken up in the enterocytes into their component parts, and they get parsed out from being micelles, which are already small, into even tinier little balls of triglycerides and fatty acids called chylomicrons. And these are small enough that they get organized into exocytosis vesicles, and they end up in your lymph in a specialized lymphatic capillary called a lacteal. And you'll get a closer up view of the lacteal in a moment. It's basically a little finger-like lymphatic capillary that reaches up into the villus. So this is all the stuff I just said. So basically we get this multiple repackaging event system where fats are digested a little bit, repackaged by an emulsifier of some kind, digested a little bit more, repackaged additionally, until we've got a small enough chylomicron that we can enter the lacteal here. So here's a closer view. Here we've got the lacteal in the center. In this picture, the capillary networks have been taken away so that you can see the transit of the chylomicron into the lymphatic system. And here's a much closer view. So, interesting difference here. Any water-based macronutrients, so amino acids, monosaccharides, etc., nucleic acids as well, those are going to enter the blood and go straight to the liver towards the hepatic portal vein. Chylomicrons also end up at the liver, but they don't get there via the hepatic portal vein. So they travel in the lymphatic system, and then they enter the venous blood in the right and thoracic ducts of the lymphatic system. So they dump into the subclavian vein, and then they circle around back to the liver via the hepatic veins. So different routes. So carbohydrates and proteins are easier to digest in some ways because they just enter the blood. And then on the carbohydrate side, carb digestion is kicked off in the mouth by salivary amylase. The stomach, there's not any ACEs really active in here. It's more the acid, if anything, that's going to pull them apart, and we get back to enzymatic hydrolysis with pancreatic amylases. And then once you get to the brush border, you're left with disaccharides. So lactose is a disaccharide, so is maltose and sucrose. So disaccharidases are released by the brush border, and they create monosaccharides that are diffused into the blood. And over here in protein territory, pepsin is the first hydrolase that divides up large proteins into shorter polypeptides. So protein digestion does not begin in the oral cavity. And then we've got all of those very, very many enzymes acting on various lengths of polypeptides that are released by the pancreas until we get dipeptides, so just two little amino acids glued together. And dipeptidases are released by the brush border of the enterocytes. And then we've got facilitated diffusion and co-transport, causing the passage of those into the blood. So that brings me to water. So I mentioned earlier that lots of water is contributed to the intestinal lumen, and that the colon is important for getting it back. It does put the finishing touches on, and if it's not functioning, you do end up with diarrhea. But the processes I just showed you, so let's actually go back one. If we're moving lots and lots of either dissolved particles, because these are proteins and carbs are water soluble, or lipids, if you think about the mass movement of particles across the enterocyte membrane during absorption, and you think about osmosis, it stands to reason that water is going to follow all of these particles back through and into the blood, because water follows solutes. Now, we typically think of that as being water following salt, but really any dissolved solute of any kind is going to be enough to create osmotic pressure, and these count. So really, reabsorption of water from your intestinal lumen is a natural consequence of absorbing your food, which is handy. So if we look at digestive secretions, so this is the amount of water that you contribute from your blood and from your interstitial spaces into your GI tract lumen. This is per day, by the way. I know that doesn't say it on the little box, but it bears mentioning that this is per day. So about 1,500 milliliters of saliva, and another about 1.5 liters of gastric secretion. So right there, that's 3 liters. That's a lot. Another 1,000 or so milliliters from bile and from the pancreas, And then your intestinal secretions from your small intestine are another 2 liters or so. So many liters per day of water you are contributing to your own gastrointestinal lumen. Colonic secretions are very small because it's supposed to be doing more reabsorbing than it is contributing, but they're still about 200 mils. And then you get most of that water reabsorbed along with your nutrients in the small intestine, so about 7.8 liters. And the colon reabsorbs the remaining 1.25 liters or so. So as ions, for example, and urobilin and stercobilin and other bile breakdown products are removed, water follows those as well. And that ends up with you, ideally, assuming you're not ill, only losing about 150 milliliters of water in feces. So assuming all of this is working properly, the system is very, very efficient. If something is not working correctly, diarrhea is a very real problem. And a lot of people don't think about chronic diarrhea as being life-threatening, but in a very real sense it is and can be. So diarrhea is the reason, for example, that people die of cholera. You dehydrate to death because you're unable to reclaim this many liters of water that you're contributing to your digestive system. So if you have cholera, the cholera bacterium inflames the bowel and prevents this 7.8 mils and the 1250 from being reabsorbed. And you basically lose water faster than you can take it in and die of dehydration. So it's a very, very real issue, and it's a medical risk that needs to be thought about by clinicians. Okay, so that ends Monday morning poop talk. Let's take a short break so I can grab some more coffee, and then we will move on to the urinary system.